So I really appreciate very much just the invitation of Dr. Vivian Stoyanov, just for allowing me to give this talk, which is probably a strange title to all of you, but I was trying to be very academic, and I'm not going to be just in details, I mean physical or chemical details, but I'll try to be much more just colloquial in terms of science. So the title of my talk is going to be focused on this issue, which is biomineralization. We are going to understand probably at the end of my talk what biomineralization means, and then what are actually the biological sensors coming from nature, and what is just the plausible use for our common life. So let me just try and start to show you the first biosensor discovered in nature. So this is a magnetotactic bacteria, so you can see here. So probably you can't see properly here, but just in this cord, the central cord, there are some minerals, some crystals, which are here composed of iron and phosphate. So those minerals allow the bacteria to move just on the earth by magnetic forces. So let me show you now what the crystals are compared to the biominerals. So in this case, we have a biocrystals. So you have crystals of proteins. So you can see here different sizes. So what is the size we are working on? So actually, you can see 200 microns. So this is the crystal of aspartyl synthetase, which is involved in biosynthesis of proteins. So you have here another type of this enzyme. This is a taumatin, which is a sweet protein. It's 100 times sweeter than the normal sugar. So this is obtained from the passion fruit in Africa. So you can see, if you take one tiny crystals, you can be sweet forever in your life, you know. So and then you have here just a cytochrome crystals, which are transferring electrons inside the cell from the inner part to the outer part of the cell. So let me show you what is the shape of these biominerals we're talking about. This is the shape of the biominerals. Sometimes it's easy to see, well, the shape is a little bit strange for us. So you can see have a kind of flights, or you can see a kind of discs. So those are the minerals. But biomineralization is just, I could not say it's a science, actually. It's a process. So biomineralization is the process involved in bone teeth formation. Even, you can see, there are different aspects. Some of them are pathological, like cardiovascular calcifications, renal or gallbladder stones, and others. So actually, what is happening in biomineralization is very interesting because all the mechanisms are controlled, biologically controlled, or they have a lot of applications. The point is, biomineralization processes are a multidisciplinary research because you need the knowledge from physical chemistry, physics, biology, material science, etc. Biomineralization process is divided into two processes mainly. So the first one is pathological, is related to renal, biliary, pancreatic stones formations, as well as cardiovascular diseases. And the second one is non-pathological process like bones formation, egg-shell formation, nacre formation, or calcium oxalate in plants. But let me show you why biomineralization is a very, very important process. So in this case, you have a biliary stones, which are solid materials usually obtained in the bile, and their chemical composition is mainly cholesterol. So please try to figure out the size of these crystals. So each large line, it's just from here to here is a millimeter. So can you imagine, as a human body, we are producing these stones inside of our body. So how to remove these stones from our body is really difficult. So the first cholecystectomy, which is just the surgery to remove these stones from the body, was performed by this guy, Langenburg, in Berlin in 1882. So let me show you the size of these stones. Can you see the biggest stone showed in this picture? This is really amazing. So actually, we are producing such a big monsters inside of our body, and you can see some stones on this hand. So this is a very, very interesting process. So actually, if we call about biliary stones, these stones are produced by the composition of the hemo group, and then thanks to the help of this hemo oxygenase, which is just one enzyme, which opens the tetrapyrrole rings, and then it's produced a biliverdin, and then this protein is actin, and then you can see there is a contribution of different molecules in the body, as well as some organs, just to produce this big and complex process. But there are some processes of biomineralization found in some different parts of the bodies. So you can see here is just an example applied to protein crystallography. Recently, a group of these people have found that polyhedrine, it's a glycosylated catepsin found in this plant, produce tiny crystals. You can see here some crystals which were shot directly in the X-ray diffraction to get the structure. So we use just the cells to produce beautiful crystals and to get this wonderful structure in situ. But let me just, to be much more precise, what biomineralization is working. So biomineralization is divided into two processes. The first one is just the biological induced mineralization, which needs actually some membranes to produce crystals. The other biomineralization process is just called biologically controlled mineralization, which is actually a highly regulated process, which produce actually bones, shells, teeth that have a specific biological function and structures. So actually, let me show you one important thing. For instance, if we are going to study biominerals, let's try to see one of the main minerals, which is called calcium oxalate in human beings. So this compound exists in two different forms: monoclinic, which has one molecule of water, and then the second molecule, which is tetragonal, it contains two molecules of water. What is happening in plants and what is happening in human beings? So in plants, you can find this calcium oxalate in the leaves, and this is just a kind of calcium reservoir, or calcium storage system. But if you look at just what is happening in main components of the stones, actually you have renal stones composed of calcium oxalate. So in our case, in human beings, it's a pathological process. Meanwhile, in plants, it's a positive process. And there are some interesting stories about these crystals. For instance, this monoclinic crystal in plants usually is forming needles. Why plants are creating two different shapes or two different space groups to storage calcium? The other point is the monoclinic crystals have a kind of needle shape. So when plants are just when the animals are eating just the leaves, they will damage because of these needles. But for instance, in the case of tomatoes, you have a tetragonal in the skin of the tomato. So what the difference is, this monoclinic form of calcium oxalate is very easy to be dissolved because plants need calcium. On the other hand, this is really complicated to dissolve because it's less soluble. Thanks to this solubility issue, these stones are not really terrible to be removed from the bodies. Can you imagine these crystals? These crystals are formed of tiny particles of calcium oxalate amorphous. So if you have a look in plants, in nature, there are 5 different types of calcium oxalate. We have prisms, drosses, raphites, styloids, and tetrahedrons. These are monoclinic, tetragonal, and tetragonal. So calcium oxalate is a variety of chemical shapes found in living organisms. And then you can see as well very important, very interesting aspects on biomineralization. So for instance, in cells, so the common cells, you can see the nucleus, the vacuola, but in plants, the idioblasts are a kind of cells which has been specialized in growing crystals. So you can see here just how the cell is growing these crystals. So you can see here the prisms, the rough ice, the dross, and the crystalline sand. So you can see the shapes. This is a scanning electron micrograph of some crystals of calcium oxalate. So you can see here the needles, etc. And then the second system, which is important in living organisms, is just the calcium phosphate biominerals. So these minerals appear into two types, hydroxyapatite and octacalcium phosphate. So these biominerals from calcium phosphate are very important depending on the organisms because they can form, in some cases, bones, teeth, and some other just parts of the bodies. So in the case of bones, the bones and teeth contain hydroxyapatite. And the structure, the chemical structure of apatite is this one. And we usually used to write this compound as this chemical compound. So what is important biomineralization? Because of this. Otherwise, we cannot survive. And then this biological control biomineralization is not only a question of solubility. It's a question of genes control. Why we don't have just three arms or two heads or whatever? This is because biomineralization is a genetic control process. As well, there are some very interesting, chemical issues. For instance, our teeth are made of this calcium hydroxyapatite. So this hydroxyapatite has this plot which is shown in here. So hydroxyapatite is less soluble at pH 7. Sorry. And hydroxyapatite is highly soluble at pH 5. So when I was a kid, let me tell you a funny story. When I was a kid, Mom used to say, don't take lemon put on your mouth. So it's going to damage your teeth. But I didn't understand properly why Mom was saying that all the time, you know? And I realized when I was just finishing my PhD that this was because of the solubility of hydroxyapatite, because lemon juice has this pH. If you take directly lemon juice on your teeth, it will be dissolved, because this is the maximum solubility of hydroxyapatite. But on the other hand, if you clean it up properly, your teeth, you brush it, you will replace hydrogen plus fluor. And then the solubility is less. So you will be protected by a chemical process immediately. So that's why you should push the kids to clean the mouth immediately if they are taking something which is acid to protect the teeth, right? This is the first message of biomineralization. The second point is this: is there any other process where calcium phosphate crystals are appearing as a pathological process of biomineralization? The answer is yes, and the process is terrible. So this is the cardiovascular diseases. So when you are just taking a lot of lipids, you like just all foods rich of grease, rich of cholesterol, so your vascular system will be like this. So this is the normal artery. When you're taking a lot of cholesterol and you're taking a lot of lipids in your diet, so the system, the cardiovascular system, will create a ring of hydroxyapatite, which will close the vascular system, the vein, to increase the pressure to remove the bubbles of grease on your vascular system. Sorry. And if the bubbles are not removed, this ring is growing, and then after closing the system, and you will die immediately. So what we're trying to understand is how to avoid this problem, so how to understand which proteins or which genes are involved in this process. So what we discover actually is when this process is happening, there are plenty of proteins which are overexpressed as a signal of emergency. So actually, when you detect that the concentration of these proteins are overexpressed in our bodies, this is just an alarm, just an alarm. Seems to be these proteins are a kind of sensors. They are just asking you for help. Don't take more food rich in cholesterol or lipids. It's very dangerous for you. So you should stop it unless you can just figure out that you will be dying in the following months because of this problem. This is not a reversible process. So the other point is, let me tell you, so in biomineralization, so we can investigate different processes trying to go through all the all the elements from the periodic table of the elements. And there are some specific places, you know, from the periodic table of the elements, which is located in the chemistry of the earth alkaline elements. For instance, beryllium, magnesium, calcium, strontium, barium, radium has been poorly explored. Why? Because if you look at just the biomineralization of silicon, you can see the structure of silicon parts in coccoliths of fossils. The most important point is just the silica has been very, very popular to form the structure bonds in the Precambrian chairs. So when we have just a reductive atmosphere, as it was in the times of maybe dinosaurs, we could be formed of silicon structures. Even, you know, the prostheses or maybe the surgery to increase some parts of our bodies are made of silicon, yeah? But this is the reason why, because silicon and silicates have played an important role in the prebiotic clay minerals used to oligomerize some RNA and DNA as well. So you see from these compounds, we can produce even nowadays by a simple experiments, some shapes coming from the Precambrian chairs. So you can see if you prepare one experiment for kids, you can prepare a gel, a silica gel from pH 8 to 10, including sodium carbonate, diffusing barium chloride through the gel by diffusion, and then you will obtain beautiful shapes which are inorganic. They are not organic. So you can see here that they look like a fly, a butterfly, and this is just a kind of alpha helix, which is completely inorganic. It's not a biological contamination. You could get these structures, and those are inorganic. So this is just the biomineralization of silica in gels. So all these chemical combinations between just minerals and some organic molecules will produce a kind of biological sensor, which is the second part of this talk. So a biosensor, technically speaking, actually is defined as a detecting device that combines a transducer with a biological sensitive and selective component when a specific molecule interacts with the biological component. A signal is produced at transducer level proportional to the concentration of the substance. Chemically speaking, it's really difficult to understand. But in a practical way, you can see that there are some molecules in nature which has been evolving through the evolution to get a kind of expertise to detect some ions, right? So the most important biosensor for me in my lab is just to investigate this compound, which is calcium carbonate. So calcium carbonate is just a compound which is obtained in nature in 3 different types: calcite, aragonite, and vaterite. If you don't know, maybe calcite is the most popular mineral. You can obtain crystals of calcite in the markets. So this calcite has a optical property, which is a birefringence property. So if you have a crystal of calcite, you can see just through the crystal, and you will see 2 images of the figure. So I don't know in here in the US, but in Mexico or in many countries in South America, if you are just in the popular markets, they used to say, this is just used to look at your aura. You know what aura means? So the esoteric people used to think that we have extra energy going around our bodies. So in the popular markets, you can find this compound, this crystal of calcite, which is having a birefringence property. It's just an optical property to divide the light into two waves, right? That's why you see just as shapes, extra shapes. Okay, so the second reason just to investigate this system from the biomineralization point of view was this bird. Do you know this bird? What this bird is? It's very popular in Central America, so probably it's not popular here because of the temperature. No, it's not the guacamaya. You know what this bird is? It's the quetzal. It's the paradise bird. Well, actually, let me tell you just the through why I'm just investigating this compound. Many years ago, part of my family were living in the south of Mexico, actually, and they paid for my traveling all the time when I was at the university. And I was in Central America, visiting many places. In Guatemala, I was inside the forest, and I saw this beautiful bird. And when I saw the bird, I noticed that it was just facing a risk of extinction. This is a beautiful bird, but it's going to disappear soon on the earth. So what I thought at that time, I'm not a person who is just taking care of the birds, but I thought if I can do something to help this bird, it should be great as a scientist in the future. So I decided to investigate how to help this bird, and I discovered that the way of helping this bird to survive on Earth was to keep the eggs protected. And then one of the ways to protect this bird is just to understand the chemistry of the eggshell formation, because everything depends on the eggshells, actually. So the calcium carbonate appears just in different systems. For instance, in the form of aragonite, just in these tiny crystals which are inside of Earth. So we can just work with equilibrium because of these tiny crystals of calcium carbonate in the form of aragonite. So calcium carbonate with magnesium is composed in this kind of structures. So it appears in amorphous materials, even in nacre. So you can see here as well in eggshells. So you can see a variety of species in nature which contain calcium carbonate. And let me tell you just a funny story. In my time at Imperial College, in the Natural History Museum, I knew that there was a big eggshell containing a tiny dinosaur. This is real. This is not a picture. So I was trying to convince the guy who was in charge of this museum in England just to give me a piece of eggshell from this dinosaur, because the proteins, the intramineral proteins which are just investigated in my lab are included in this mineral part. So I was trying to get a little piece of this dinosaur to investigate what was happening at the time of the dinosaurs in these proteins. So of course, the person said, well, I think it's really complicated, Dr. Moreno, in a British style, because this is perfectly closed, and it has been closed since the last 100 years. Unless you can break the glass, which is this size, we can give you this, but the police will come immediately. So I said, okay, thank you very much, I'm not interested anymore. The reason why I was interested in this piece of eggshell is because actually the closest animals to the dinosaurs are these. So actually in the southern part of the earth, actually in Australia, even in New Zealand, you have these birds. You know what these birds are? This is the ostrich, the Rhea americana, and the kiwi is just something like this, okay? Those animals, those birds, avian, are coming from dinosaurs. So what I was trying to do, actually, is to investigate which proteins are related to calcium carbonate biomineralization. If you look at the eggshell, if you cut the eggshell, first you will have a membrane of organic membrane in here, and then you will have calcium carbonate in this area, and then you will have a polysaccharides onto the top of this eggshell. So these polysaccharides are protecting the eggshell against bacteria or fungi, whatever, right? And then if you just dissolve this calcium carbonate with acid, acetic acid or whatever, so you will extract proteins directly inside the intramineral part of the eggshell. So those proteins are coming from this time. So what is interesting is, okay, if that is true, what is going to happen? So I have plenty of possibilities. For instance, in terms of eggshells, I can take eggshells from these birds, from snakes, from turtles, from different animals, from hen or goose. What is the best to start this research? Because this is going to give me just a kind of information from the dinosaurs, from the beginning of the earth. So you can see there are different types of eggs. So you can see even a variety of colors. You have this color, just brownish, yellow from the ostrich, from the Rhea americana, this color. So we decided to start with the hen at the beginning. And what we did actually is we were trying to dissolve the eggshell in acid. We extracted proteins. We discovered that in the eggshell formation there are kind of proteins, the intraminal proteins, which are involved in this nucleus formation. And then we started to understand the process by saying, okay, there is a membrane, organic membrane, which is a kind of template to allow the crystals of calcium carbonate to grow. And maybe this process is controlled by a protein, which is called, in the case of hen, it's called ovocladin. In the case of ostrich, it's called strutiocalcin. So in each animal, in each avian, you have different proteins. Which is very interesting is in the eggshell of ostrich, you have two proteins, intramineral proteins, which are controlling the biomineralization of calcium carbonate. In Rhea americana, there are two. In Dromaeus novohollandi, you have dromaeocalcins 1 and 2. But in the hen, you have only one protein as well as in goose. So the reason is all these proteins are related in terms of the structure, in terms of the contents of the amino acids. And then if you like to understand the process, you can compare it in the sequence of amino acids. You know, a protein is just a complex polymer constituted of 20 amino acids. If you just compare what the composition of amino acids is for different species of, these birds, so you can see there are some areas which are pretty similar each other. So if you isolate the proteins, you will see a very interesting process. So most of the proteins which are selective for calcium in nature, they look like this. They have this structure. This is the group of proteins are called lectin C-type proteins, and the structure is like here. So they are trained by nature just to select or to work as sensors of calcium, and they put calcium in here, in here. So this is the structure of ovocladin 17, which is a protein, intramineral protein found in these hens, in eggshell from hens. You can see the structure is pretty similar, but this protein is not selective for calcium. Then the answer is, well, maybe this protein is different because it was trained by nature for a different activity. So we were trying to introduce calcium in this protein, and calcium was just nothing. It was just not located in any parts of the protein. We solve the structure of Oclein from eggshells from hen. We solve the structure like this. We look for the places where calcium could be located. We didn't find calcium. And we thought, well, maybe this protein was not trained by nature to select calcium as a biosensor. Maybe this protein was trained for something else. And then we decided to say, well, we are going to isolate this protein just by different chemical and biochemical methods. And then we separated two proteins from the ostrich eggshell, estrutocalcin 1, estrutocalcin 2. And we were thinking that these proteins should be sensors of calcium, but it was not true at all, because when we prepared just some electrochemical analysis by using these devices to select or to investigate the concentration or the selectivity for calcium, we detected that these proteins were selective for carbonate ions. What is very interesting is by using dynamic light scattering methods, which is a biophysical method to detect the size of macromolecules in solution, we detected this very interesting process. So these proteins in water, they are highly aggregated. You can see this just plot shows different sizes of the proteins, isolated proteins in water. And then if you look at just this structure of the protein and the nucleus which are forming the eggshell, so you can see by light scattering different behaviors of these proteins, different aggregations. But when you just take this process and then introduce in the fluid, in the uterine fluid, the concentration of calcium is about 10 millimolar and carbonates is 70 millimolar. You just introduce the protein inside the dynamic light scattering apparatus, and you investigate the aggregation stage at different concentrations of carbonate. You will see here at 10 millimolar of carbonates, you will see that there are plenty of aggregations. At 70 millimolar carbonate ions, so the system is really regular, it's homogeneous. So at higher concentration, it's not. Maybe this is related because in the uterine fluid, these proteins were trained to select carbonate. So this is happening for different proteins. This is the SK2. You can see at 70 millimolar is not selective for carbonate as the SK1 is, but this is very selective. So you can go through angiocalcin, which is just obtained from goose, and then this protein is 10, it's a solution 10, 70, and 100 millimolar. This is not really selective. This is the case of lysozyme, which is just not intramineral, and then we can find that there is no selection of carbonate ions. Okay, so the message is these proteins used for nature just to select carbonate instead of calcium probably are doing some work which is very, very specific in combination with proteins. So you can see this protein, estrutocalcin, is selective for carbonates. Once you get the structure, the 3D structure, you will be able to produce or to just to prepare the mechanism for this interaction. And then the proof, the chemical proof, to see whether these proteins are just a sensor of carbonate is this. If you crystallize calcium carbonate in the lab, so you need only to put in these droplets calcium chloride plus ammonium carbonate in here, so you can produce beautiful crystals of calcite. But if you contaminate the system with different concentration of proteins like estrutocalcin 1, estrutocalcin 2, vocladin from hen eggshell, and lysozyme from eggshell, from the internal part of the eggshell, you will see that the system, when the system is contaminated with small amount of this protein, the shape of the crystals is transformed. So you can see at 50 micromolar contamination, 100 and 250 micromolar. So the shape of the crystals is completely different compared to the control. You can use SK2 as well, and ovocladine as well, and finally lysozyme. So you can see these nucleus which are obtained in the eggshell formation just from the beginning are very comparable to these nucleus which are found in the contamination of the system by these proteins. So when we solve the structure of streptocalcin and we compare the structure of this protein with the ovocladine, so have a look. The difference is almost nothing. So both proteins are completely similar. So the question is, why nature has trained the same protein in a different way? For instance, in ostrich, the eggshell it's not very tiny like in the hen eggshell. So you can break the eggshell without problems. But meanwhile, in the ostrich, it's like a ceramic. So the question is, why nature is training proteins to be sensors of calcium carbonate to produce these beautiful eggshells? So what really we need to know to understand this process, this biomineralization process of calcium carbonate biomineralization, is not so much. The knowledge is minimum. What you need is just to know something about precipitation or crystallization. So the chemical control is just to know something about solubility of proteins, solubility of minerals, concepts of supersaturation, nucleation, and growth of crystals. So what is happening, the final answer, I mean the real answer, is coming from the chemistry behind the biomineralization process. Most of the minerals I've been trying to show you along this talk are related to these values of solubility. They are showing a minimum solubility. So you have the case of calcium carbonate in the form of baterite, aragonite, calcite, calcium phosphate, as I mentioned in the cardiovascular diseases. So the solubility is minimum, amorphosilica, some oxides of iron, etc. So let me just almost to finish, sorry about just this disturbance of the computer, but you know, when we all know that crystals nucleate and grow from saturated solutions. If we like to obtain crystals, what we need just is to put enough amount of solid inside the solution to oversaturate the system, and then the solid state will come out immediately because of supersaturation processes. So this is the normal way of obtaining crystals in vitro, but nature is doing exactly the opposite way, because biology has chosen another pathway. Crystals are grown from an unstable solid colloidal phase, almost devoid of water in nature, which is very interesting for all of you, because what would happen on Earth if water would be not really available in the future? Seems to be that we could survive even having less amount of water. This is a very, very important message. Nature has protected us to survive. And even the idea of my talk to show you very simple issues like investigating proteins, intramineral proteins inside the eggshells, is to show you that there are many, many proteins in nature which has been specialized along the evolution to do many, many biological functions. They are working, you know, as a biosensors because they are just detecting some ions to produce biological functions, or they are creating minerals to protect or just to construct our bodies. The point is how nature has specialized genes to control this process. The answer is open. Which genes are controlling this process? We really don't know nowadays. So many animals do not form their crystals directly. They first produce a transient precursor mineral phase that subsequently transforms into the mature phase. This concept is very simple, but this is very interesting. So many things we learn at the university level are not true at all. Because when you are just investigating many biological processes, you will be surprised. It's not simple chemistry, it's not simple physics, it's not simple biology. So you need to be a multidisciplinary scientist to understand all of these processes. And my simple discovery, you know, to see this beautiful bird from the forest in Guatemala, the quetzal, showed me just how limited I am as a scientist to protect these birds. It's not very easy to do, because actually you can produce knowledge to produce strong eggshells. But even if you control the process, you can produce a kind of stone of eggshell. The bird will not survive, because the shell should be broken, you know, by the bird. So there is a compromise. You need to protect the bird, but not in an extreme way. Otherwise, it's exactly the opposite effect. But on the other hand, if you are just in a places where there is no water, so actually the eggshell is not formed because there is a deficiency of calcium, and there is no calcium available to form these eggshells. So this is a paradox, actually, because nature is trying to survive, even producing a solid phase, almost in environments devoid of water. But if there is no water, there is no eggshell. If there is no eggshell, there is no life. So this is a very interesting issue in nature. So I would like to say as well that in terms of biosensors, nowadays in my lab, we are very much interested in some proteins which are just, or were trained by nature in transferring electrons. I'm not going to show this in my conference. These proteins are just the cytochrome C, which are the proteins I showed at the beginning in red color. Do you remember? So, okay, those proteins were trained by nature to transfer electrons. Since many years ago, we have started to understand the process by trying to solve the structure of this cytochrome and understand at the same time how the transference of the electrons is happening in the internal part or the external part of the cell. But this process, as simple as it is in terms of transference of electrons inside a membrane, a biological membrane, it's a very complicated issue, because this protein, cytochrome C, is not very easy to crystallize. So this protein is very sensitive to changes in temperature. So if you really like to purify this cytochrome C to investigate this electron transference, once you isolate the protein from the original source, the protein will be damaged. And it seems to be that if you don't have the ability, the biochemical ability to purify proteins, so you never will know the structure of this protein, and then you will never get information about the transference of electrons. So the idea of investigating these processes is because maybe in the near future, we are going to get solar cells based on proteins, or based on biomimetic elements remaining these proteins in the case of transference of electrons in nature. And there are many examples of these biomineralization processes which are very important, not only in material sciences. The most important processes are just in biomedical applications. How to understand the cardiovascular diseases is just a very big issue. Because actually, what we discover is when we have facing problems, this cardiovascular problem, there is a protein which is overexpressed when you have a problem. It's just osteopontin. If we could get a system, a biosensor, sensitive to detect this osteopontin, any part of your body, even in the finger, just detecting the amounts of osteopontin overexpressed by your cells, you are facing a very high risk of getting cardiovascular diseases. So this is another example why biomineralization processes are very important to know. And the other point is, for instance, is why nature is controlling, genetically speaking, the size of my hands. Why I don't have 3 instead of 2? So why nature has trained genes to say, well, so calcium phosphate is the main component, in the form of hydroxyapatite, it's the main component of my bones, right? So if I'm playing football, soccer, or American football, I can break my leg forever. And as a scientist, simple scientist, I could say, well, why I don't replace my bone with calcium phosphate? It's the main component of the bones. The answer is not as easy as a chemical compound. So biominerals are a mixture of pure chemicals plus something else, biological. So our bones are a mixture, a complex mixture of calcium phosphate plus proteins plus polysaccharides and DNA together. So this mixture just produce specific mechanical properties to allow the bones to be a little bit flexible. If I could just replace my bones with calcium phosphate in crystalline form, it could be breakable easily, because this is a solid. But when you include proteins or some biological macromolecules, the properties will be different. So I like to finish this talk. Sorry about the disturbances again of this computer, but I like to acknowledge my people, and then first I like to conclude that if we really understand what is happening in nature in terms of biomineralization processes, we should be aware that there is no simple chemistry behind, there is no simple physics behind. So people who are involved in biomineralization processes need knowledge from physics, chemistry, biology, even mineralogy, all together to understand biological processes, right? And our origin actually as a human beings or even as a living organisms on Earth basically was coming from biomineralization processes. We are surviving on Earth because of these processes. Because the biomineralization of silicon produced the first polymers in nature to produce RNA. And RNA was the catalyst of life. Without catalyst of life, we cannot produce DNA and we cannot produce complex systems. So thank you very much, and I really appreciate just the invitation of Vivian Stoyanov to this lecture. I'm so sorry the problem is still there, but I don't know, maybe I was proud of being Mac user, but maybe I will switch to another system again, maybe not. And I really appreciate very much if you just ask me any questions, just any curiosity based on the topics I was trying to convince you that biomineralization is important in nature. Thank you very much for your kind attention. Yes. Okay. Well, it depends on the eggshell. For instance, in the ostrich, you have just for 100 grams of eggshell, 1 gram of protein. But on the other hand, from the eggshell from hen, so the amount is minimum. So if you like to isolate the protein from the natural source, it's really complicated because the process to purify the protein to get highly pure protein to get the structure takes a long time, and the amount of protein is minimum. So it's much better if you just isolate the gene which is involved in overexpression of this protein, and you can produce amount of this protein to investigate the structure. Okay, it's minimum actually. Sorry, say it again because I can't hear you properly. Yes. Well, selecting-like proteins were trained in nature to identify calcium. The point is, when you compare the structure of those proteins, lectin-like proteins, with structurecalcium proteins, the intraminal proteins from the eggshells, the structure is almost similar, the shape of the structure, but the function is different. That's why the question is it's very tricky. Why nature has prepared two types of proteins with the same structure having different function? One is for selecting calcium, and the others from the eggshells are for selecting carbonates. This is a very tricky question. At the moment, I don't know. I don't know. Okay? You're welcome. Yes, I mean, there is a science which is called biomimetic, and nowadays we are learning from nature. So, for instance, engineers or physicists who are involved in material science, they're trying to understand these processes. Because actually, if you try to take just a piece of eggshell from ostrich, and then you try to break it, it behaves as a ceramic. Strong ceramic. So probably in the near future, we will have a kind of replicas of many compounds made by nature in our common life. So actually, nowadays, not only just the ceramics obtained from this information from the biomimetic of the eggshells from ostrich, now we have probably devices, electronic devices, which were a copy of devices made by nature many, many years in evolution. This is just the answer. We can do that. We can do that.
Another question is related to the first part of your talk, which was like you have all these deposits that
The silicon. This is a very interesting question. It was actually one of the PhD thesis questions from my student in my lab. So actually, we're looking for the protein or just the chemical compound to inhibit this process at the beginning. So for instance, we were looking for the possibility to detect osteopontin when this process is starting, and then we were trying to look for the way to stop the process in situ at the beginning. But the point is, it's very complicated because it's not just only the overexpression of one protein. It's much better just to look for the gene and stop the gene to stop the process. It's much better to stop the gene more than to inhibit most of the proteins. So nowadays, the related question, the related answer to you should be how nowadays the diabetes mellitus is just stopped. It's not the proper medical treatment in the future is not going to be just by drugs, but treatment of drugs. It's going to be by the use of genes. So you can just modify the genes, correct the errors in the genes, put the genes inside the cells by using just virus introduced to the bodies, and then you can correct the error, biologically speaking. So maybe the answer should be not just directly including inhibitors. Maybe the answer should be to understand these processes, biomineralization process, in the basis of genetics. This is my guess as a scientist. Okay? Is what, sorry? I haven't tried this property, but should be. It's amazing material.
Once I heard that it's iridescent because the arrange of the crystallites are like a metamaterial. So it is also a calcium carbonate. Why it grows like this, like a metamaterial or another?
Well, it can be like that because actually, if you look at from the first part of my talk, we have different colors of the eggshells. Do you remember? There were yellow, brownish, black. And what exactly is the main function of the black eggshells, for instance? Is this because of protection? Is this because of something else? Maybe Roberto knows that because he's a biologist. Perhaps. So there are many answers. So, for instance, when we discovered these properties at different colors, my main question at that time was, why this eggshell is black? And we were trying to isolate the chemical compounds from this Rhea americana eggshell, and then we didn't discover anything different. It was just color with some chemicals, and that's it. But there are not many other properties. Maybe it's just to protect the eggshell against changes in temperature, and maybe that is all. But in your case, in the ostrich eggshell, that property should be very interesting, because those birds are living only in the southern part of the Earth. They are not existing, although we have Rhea americana, but it's not americana from the US. It's Rhea americana because it's in the southern part of America. And then the ostrich is only available in Australia. And the kiwi is only available in the southern part of Australia, in New Zealand as well. So what the reason is to have those birds living only in there? Why? I really don't know. It's very interesting. Whilst many birds are flying all over the earth, anywhere. So it's a very interesting issue, okay?
So if there are no other questions, let's thank Professor Moreno again.
Thank you very much.